Hello everyone. So in this session we are going to learn about some important concepts regarding to the principles of inheritance and variation. So in the last session as I told you about the contrasting characters of pea plant, I have written again on the board, you can check it out once again. So we'll repeat once again all the terms and what we studied in the last session. So the first of all I would like to tell you about the definition of inheritance. It is a process by which the characters passed by or passes from one generation to other generation, from parents to offsprings. So it is known as inheritance. So and then after that we studied about the variation. Variation in sense, variation is nothing but it is a degree by which the changes occurs from parents to offsprings. We never resemble to our parents. Okay, so that variation, that changes is known as itself variation. So after that we studied about one scientist. Who is that? He was the father of genetics. So the father of genetics is Gregor Johann Mendel. So Mendel, he chooses the pea plant Pisum sativum for his experiment. He worked on the same plant for 6 to 7 years, and he told about the inheritance, the characters inherit, it used to inherit from one generation to other. The same thing happens in every human in every organisms. Only those organisms which reproduces by means of asexual method. So after that I explained you about the 7 characters. So before starting the chapter, before starting the monohybrid and whatever is there in your NCERT, I would like to mention few terms, few important terms related to this chapter. If I start telling about the genes and about the homozygous, pure lines, everything is there. Before starting those concepts, let me introduce you, let me tell a few lines regarding to the terms, so that you can catch easily when it comes in the concepts. So with this we will start with the important terms. Now let me tell you about the 7 contrasting characters. The first character we learned on that day is stem height. Of what? Stem height of pea plant. If the pea plant is tall, so if the pea plant is tall or it is dwarf or tall. So these are the 2 characters we used to study in this chapter. So by describing these 2 characters regarding to the height of the plant, and likewise flower color, I have written on that day, I guess purple it is, no problem, whatever it is, it may be. Violet is the dominant one, and for the dominant flower color, the recessive character is white. So dominant and recessive in sense, what is dominant and what is recessive? So dominant is the character, or this is the gene, when it comes in the organism, when it comes in this pea plant, either tall or violet or axial, the recessive character do not expresses. It do not allow the recessive character to express. It will suppress the recessive character. Only the dominant character will be in appearance. So likewise the dominant and recessive characters will be, and flower position, axial is the dominant character and the terminal position is will be the recessive. Pod shape, So pod means a complete pod, what you used to peel that pod, no. So that pod, if it is flattened, inflattened, that inflattened pod shape will, it indicates the dominant nature of the pea plant or dominant nature. If it is constricted means it is a recessive. When the recessive characters expresses only in the absence of dominant character. If the dominant gene is not there in that plant, then only the recessive character it used to appear in the plant. So pod color is green is the dominant one, yellow is the recessive one. Seed shape, coming to the seed shape, if the seeds are round, those are considered as dominant nature. If it is wrinkled right in the pod itself, then it is known as, or it is considered as recessive. Seed color, dominant is yellow, green is recessive. So with this we will start with the some concepts. The first one I would like to tell you about the gene. So on that day we discussed about the genetics. What is genetics? It is a branch of biology which deals with the study of, which deals with the inheritance and variation from one generation to other generation. So that is what the genetics. It's a branch of biology. It comes under biology. So after knowing the concept of, after knowing that genetics and inheritance and variation, so today actually in actual term we are learning what is gene. So what is gene? It is just a unit of inheritance, a functional unit of inheritance which brings changes from one generation to other generation. If I am having a gene of my parent, I am expressing my parent character. So that gene inherits from my parents to me. So the gene, what it is doing, it is passing from one generation to another generation. So the actual concept, the definition is, it is a unit of inheritance. Unit of inheritance. So this is the what is that? They used to ask you in the examination, what is gene? So if you write unit of inheritance, that is sufficient, more than enough. So in the earlier days, when the Mendel described about the inheritance or about the heredity, he used to, or he took the term as factor. He didn't said, or he didn't coin the word. He just told some factors are there, some units are there that inherit from one generation to other generation. He used to name those units as factor. But gene later on, Johansson, he coined the term from factor to gene. So in total, according to Mendel, factors are nothing but gene. Okay. So next is, after the gene, we'll study, likewise I'll tell you some concepts on the terms and their definition. Terms and their definition. So only few of the terms they'll ask you in the examination, not all. Allele. So allele is nothing but, so as I have written on the board, tall, dwarf, violet, void, axial, terminal, what are these? These are the dominant and recessive characters, a describing character, right. So here allele, these are the two forms of genes, forms of gene. two forms means, for example, dominant and recessive. By taking only any one of these contrasting character, I'll take any one character out of 7. I'll take the height of the plant or height of the stem. What it is indicating, the dominant nature of the stem height is tall and the recessive one is dwarf. I'll write here the tall and dwarf. I have just taken the example to describe about the allele. Okay, so alleles are nothing but forms of gene. So normally we call two forms, it will be two forms, it will be in two forms. What actually it is? It will be dominant one and it will be in a recessive one. So these are nothing but alleles. Alleles are forms of genes. And then let me tell you few more concepts related to that. The tallness, when we used to cross all these characters, when we used to come to the what is that, some schematic representation we used to put in this chapter, describing the parent nature and then offsprings and then next generation and again some other next generation. So overall in that schematic representation we never used to write tall, dwarf. Here to describe the tallness and to describe the dwarfness of the plant, we used to give the alphabetic English alphabets, the small alphabets and then the bigger one. So like for example, we have taken the stem height to explain or to tell about the allele. To explain you or to describe about the allele, I have taken one example here out of the 7 characters, that is stem height. Stem height of the pea plant. What is the describing character? What is the describing character for dominant nature? It is tall. The describing character for recessive nature is dwarf. Okay. To write the tall in short form, we use, as I told you, we used to write in a English alphabet. So that is capital T. Capital T indicates the dominant nature of the plant. And likewise here dwarf also there. Dwarf describing word also there, right? So to write to indicate the dwarfness of the plant, don't write the capital letter D. So this is incorrect. This is wrong method of describing the character of dwarf. Whenever we started describing or we started writing the capital letter to describe the dominant nature of the plant, when it comes to the recessive nature, just write the small fonts of the same letter, like small t, small font. Dominant, capital T is for dominant, the same letter small t, small font is for recessive nature. So these are the very important, you have to understand this. When I start writing capital T, small t, if you don't understand what I am telling you, what I am describing, what I have taken the example, you never come to know about the principles of inheritance and variation. So like I have written like that, okay. When it comes to the flower color, what shall I write? On that day I have written the purple color or violet color, whatever it may be. Just write the capital V and for the dominant nature and small v for the recessive nature. Likewise, flower position, axial. Axial is the character indicates the dominant nature of the plant. Axial, capital A, terminal, the same letter, small font, small a. Pod shape, inflated, capital I, small I. Pod color, green is the dominant nature. So don't get confused with the pod color and seed color. Okay, pod color, green is the dominant one, yellow is the recessive one. So green, how you are writing the allele, capital G. This is the letter which tells about the dominant nature of the pod color of pea plant, and small g will be the what? Recessive gene. And seed shape, capital R, and wrinkle will be the smaller one, small r. Seed color, capital Y, green dominant will be capital Y and recessive will be a small y. So this is how we are going to write. It will just tell about the contrasting character, either it is in a dominant position or in the recessive position. So we will see the next term. Whenever I wrote capital T, it means it is indicating the stem height or heightness of the plant. Capital T indicates, always it indicates the dominant nature. And then one more thing is there, multiple alleles. Multiple alleles I will tell you later. So once at one time we can describe two characters like stem height and flower color. 7 characters are there. We never used to describe all the characters once for all. First 2 character or 3 characters and then next and the next we used to like to check the offsprings. When we have crossed one plant, a dominant plant with the recessive one, to check the offsprings, either it is either it is having its parent character or dominant character or not. To check that we used to take any 2 characters of the plant, and then after the allele, that is the second one, the second term. So the third one will be the homozygosity. So homozygous condition. Homozygous condition is, what is that? Homozygous condition, if these genes, whenever we used to write the gene, we never used to write in a single letter. We used to describe a gene, or we used to write the gene in pair. Capital K, capital T. Dominant one, small t, recessive one. Okay. Recessive is the character which do not express in the presence of dominant gene. Dominant character is the one which suppress the recessive character. Okay. Always the dominant character, it will express in the organisms. Homozygous condition. So homozygous condition, just understand this. Homozygous condition, here I have written the letter 2 times. It indicates it is in a same fonts, capital T or small t. I have not mixed the letters like this. Okay, if I mix these letters, what will happen? I will tell you later. So this is also one term I will tell you. After the homozygous condition, heterozygous condition. Right after homo, we are going to learn about the heterozygous condition. Homozygous condition, so this is the condition where the genes are similar in nature. The genes are of same type. If in the organism, we'll take this Pisum sativum itself. Alright. If in this Pisum sativum, it is having a gene, that gene, it is of same type. Same type means, consider one gene, we are having that gene describing the height of the plant. If the gene having one more of similar type, why you are telling similar type, why not dissimilar? How come this dissimilar will be? That I will tell you in the heterozygous condition. Just understand here, if the gene is of, if these genes are of similar type, same type, this condition is known as homozygous condition. Okay, and so capital T in sense homozygous, capital T, how we can express, how we can write the appearance or characters of this letter. So capital T indicates the tallness of the plant, tallness, height of the plant. Alright. So for the tallness, I have written, I am just giving the example by taking capital T, small t, small t. It is not compulsory to explain only by taking the height of the stem. We have to explain the homozygosity, heterozygosity, that is not compulsory. I am just taking the example. So capital T, if the genes are of similar type, it means the gene is in homozygous condition. What it is indicating? It is indicating the tallness. How we used to describe this capital T? This is homozygous dominant. Homozygous dominant. Coming to the small t and small t, these two are of similar type but recessive nature. Homozygous, homo means similar genes, right? Okay, so homozygous but small t, small font always indicates the recessive nature of the plant. Homozygous recessive, homozygous dominant and homozygous recessive nature. If both the letters are of same font, it will be homozygous dominant or homozygous recessive. Okay. So after the homozygous condition, we are going to study about the heterozygosity or heterozygous condition. Heterozygous condition. Hetero, the name itself saying two dissimilar. What dissimilar? If one is dominant and another one is recessive, like dominant one more will be recessive one. These two are dissimilar genes. Alright. So if in an organism of if it is describing one character, if one gene is describing one character, those genes are in dissimilar manner. One is dominant one is recessive one. These two different genes are known as a heterozygous gene. This condition is known as heterozygous condition, and need not to explain or need not to describe the nature. Why? As I told you, whenever the dominant character is there in an organism, that only expressed. The recessive will not, it do not express. Why? Because this dominant gene suppress the recessive gene. So, while describe, as I told you, here homozygous recessive, homozygous dominant. But here need not to write the dominant one. It is understanding. Wherever the capital T or capital letter is present, side to the small one, there in that condition, or in total in the heterozygous condition, it is understandable. Heterozygous dominant one, that will be a dominant one. Heterozygous dominant. And with this, hope you have understand this concept. If you are not getting my concepts, please ask me. The next one is, after the heterozygosity, the next thing, I'll write here itself. The next term is, fifth one, phenotype and genotype. Phenotype, like what I have written on this board: tall, dwarf, violet, white, axial, terminal. These are all the phenotypes. It means describing letter or describing words are known as phenotype. Describing words in the organism, if we are describing any character, like stem height, it is a tall, dwarf, flower color, violet, white, flower position, axial, terminal. So these are all the phenotype of an organism of the plant. Phenotype is a physical appearance. Describing words are physical appearance. We can see the tallness of the plant, right? We can see the dwarfness of the plant. We can see the color of the flower, or we can see the position of the flower. So whatever the characters it is appearing, it means it is known as phenotype of the plant or the organism, the physical appearance. And then what is genotype? The character or genotype, the name itself saying gene type. The genotype is nothing but the genetic makeup of the character. Easily we can, by looking at the plant, we can tell this is tall plant, so that is the dwarf plant, the flower color is like that, but we can't make out the genes of the plant, whether it is in a homozygous condition or whether it is in a heterozygous condition. Whatever the gene it is having the plant, that gene indicates, or that makeup indicates about the genotype of the plant. How to calculate, how to check the genes are homozygous or heterozygous? By cross checking, by crossing with the pure line. Well, I'll tell you in the next sessions, I will come to know how to check the phenotype of the organism. So after the phenotype and genotype, the next one will be cross. So the cross is nothing but taking two plants, though we are in Pisum sativum, we are going to cross two plants, like one plant of stem height will be long, the color of the flower will be violet, and like that we don't know what characters we are having in one plant. one more plant we are having, like it is dwarf, or the color of the flower will be white, whatever it may be. So when we cross these plants, what will have? Will be having the offsprings of it. So that process is known as crossing. Crossing means reproducing the two plants. We are reproducing again and again, reproducing the offsprings by taking two plants. So that term is known as cross. The seventh one will be filial. Filial is nothing but, think we have crossed one plant, one parent and one more parent. Okay, I am just taking two plants. Consider we are having two, let me write here, male plant, female plant. You can ask me male plant, female plant, it won't be there. The flower is bisexual. Likewise you can ask me what we are doing here. Male plant in sense, by the method of, we are applying the emasculation method. If we need only male plant, we are taking out the female or the gynoecium, and in the female plant in sense we are taking out the anthers. Okay, so by doing the emasculation, we can have only male plant and female plant, describing some different characters. So when we cross the male and female plant, or any two plant you consider, okay, it's not compulsory we have to cut the anthers, we have to take out the ovary. We are just describing this, no, we are just understanding this, no. So you can think like that. So male, the plant, two plant we have taken. When we cross these plants, what the offsprings we get from these plants? Some offsprings will definitely get the offsprings, the plants, the next generation. So this generation, the first generation, is known as first filial generation. The selected plants are the parents. The first obtained offsprings are known as F1 generation. Later on, again we are crossing between these two or any other plant, or we can, if we are selfing this plant, selfing means self pollination. Okay, so the whatever we obtained from the F1 generation, those offsprings are known as F2 generation. And again, if you are going to cross these two plants, F2 generation offsprings, what we get in the future, the offsprings will be considered as F3 generation offsprings. Like that, we can continue crossing the plants up to 6th generation. What is the use of it? I'll tell you. That is also there in the concept. That's why I'm telling all this. Filial means whenever I write F1, consider that is filial. F1, first filial generation, second filial generation, third filial generation. Okay, so after that, after the filial generation, the next is Punnett square. Punnett square. The term, this concept named after the scientist known as Punnett. So what is Punnett square? The Punnett square is the square, or it will be in a box shape. What it indicates? Why it is there in the principles of inheritance concept or in this chapter? When we cross the parents, I'll tell you one more example here, taking the tall and dwarf nature. Consider I am crossing the tall plant with the dwarf plant. What I have written here? I have written the genotype of the plant, saying homozygous dominant, homozygous recessive, homozygous tall plant, homozygous dwarf plant. Okay. When we cross these two parents, these two plants, what we'll get? We'll get something, some character, some offsprings will get. The offsprings will be having it will be having the character from its parent itself, right? To check the offspring's character, we used to put the square, Punnett square or checkerboard. It is also known as checkerboard. It is there to check the genotype and phenotype of the offspring. So, before taking the Punnett square, we used to select, first we have to select the parents. The parents are in the homozygous condition. Okay. The first one of the parent, consider this is the male one. Here you have to write the genes of the parent side by side, like that it has to flow. And one of the parent, this next parent genes, it has to write here. You have to write like this. Alright, so the genes are separated now. Alright, so when it gets separate, when it crosses with the one more parent means here one more parent is there, no. When it get crossed, when these two genes mingle, when these two genes crosses with one another, the offsprings from the parent, what we are having in the first generation, what we are having as an offspring, those offsprings genotype will be, how it will be? I'll write that one, T and T, capital T, small t, T and T, capital T and small t, T and T, like this you have to cross, like this you have to write, taking male parent this side, female parent this side, you just cross it, cross the genes, capital T, small t, capital T, small t, capital T, small t, and the same thing. These are all the offsprings. Capital T, small t, capital T, small t, capital T, small t, capital T, small t, these are all the offsprings. Now, you can write the phenotype and genotype of the offspring now. Capital T, small t, what it indicates? This is the heterozygous condition. Heterozygous condition and heterozygous condition, and need not to write the dominant or recessive one. Whenever the heterozygous condition appear, it is definitely no need to write, it means you have to write, but it is understanding, it shows the dominant character. Heterozygous tall plant, all the offsprings will be of tall, the stem height, the stem length will be tall in nature. So, from where we got these offsprings? When we cross with the male, dominant homozygous parent with the dominant recessive, homozygous recessive parent, the offsprings will be 4. Why you have written 4? Only 4 offsprings. How much you can obtain the offsprings? Only to check the pheno and geno of the offsprings, we used to cross by putting the Punnett square. So that is what about the Punnett square or checker board. The next term will be selfing and pure line and monohybrid, dihybrid and trihybrid. So selfing, in sense, selfing is simple, it is. So here what we have in the offsprings, only this offspring, this plant, if we self-pollinate means the gynoecium to the stigma, if it is a stigma, if we dust the pollen grain on the of the same gynoecium, a flower, in the flower gynoecium and androecium will be there. So what we are doing, we are not allowing any other pollen grains to sit on the stigma of the selected plant. We are self-pollinating the plant. In that condition, what we have to do, we have to take the anthers or pollen grains of the same plant, of the same flower. We are dusting the pollen grains on the stigma. What we are obtaining the next generation. So that process is known as selfing. And then pure lines, is the term, pure lines, to obtain the purity of the gene. So we can see here, it get mixed. We don't want this recessive character, only we need the pure homozygous dominant character. What we have to do, we have to obtain that pure dominant homozygous organism. We have to self-pollinate the plant for generation, for 6 to 7 generation. Why? To obtain the purity. Purity indicates the dominant one. Purity indicates the better yield qualities. To do that process, to follow that process, to get the purity of the organism, to get the purity of the genes, when we start self-pollinating the organism for generation to generation, at the end we can obtain the pure line organism. If you didn't get the pure line, it will again come in the concept. There you will get what I am telling. Everything is going to repeat in the coming concepts. But when I start describing or start writing the phenotype, don't get confused or don't be dumb without knowing these concepts. You can't put the checkerboard or you can't solve that parents. Okay, so let me tell you one more thing here. The next is monohybrid, dihybrid, trihybrid. So this will be the 12th and 13th one. Monohybrid, dihybrid, trihybrid, these are the hybrids. Hybrids means crossing the parents. So if we are selecting one parent, it means each and every characters will be there. But we are concentrating any one character of these, out of these characters. Any one character means we want only tall plant. We are not interested in flower color or any of these characters. Concentrating only one character, and to get a purity of that character, when we start crossing the plants with the homozygous dominant or homozygous whatever it is there, so that character, only taking one character, that indicates, or that process is known as monohybrid. if we cross a plant and if we are crossing checking only one character, if we are checking only one character, it is known as, this hybrid is known as, this crossing is known as monohybrid cross. Monohybrid cross means only tall or tall, not or, tall and dwarf, describing only one character. One more example, if we take describing only one character means only violet or white. Describing two characters, it means what? Describing two characters means tall and violet. We need a tall plant with violet color flower. To get two characters, when we start crossing the plant, so that crossing or that hybridization is known as dihybrid cross, taking two characters. Considering two characters, if we cross the plants, it is known as dihybrid cross. Likewise, you can, so this is the dominant one. If we cross with the recessive one, what will be? Tall, dwarf, violet, white. And the same thing, tri-hybrid means taking three characters. Tall violet axial, dwarf white terminal. Tall violet axial and dwarf white axial terminal. I am writing this side by side. Why? Because these characters indicates the dominant nature, these character indicating the recessive nature. So, few more concepts are there, terms are there. Let me tell you when the concepts come, when the contents come. I'll keep on telling you all those, like multiple alleles I forgot to tell you. I'll mention in the upcoming content. With this we'll end this session. In the next session we'll start with the monohybrid cross. So, before starting that concept, I'd like to repeat one more time, all these terms are important. Just learn it, understand it. When you understand these terms, then you will become, then only you will come to know what I am telling you in the monohybrid or dihybrid. Thank you.